Good afternoon, everyone, and welcome to the live session for Biology GCSE. I'm going to start things off today by, well, it's just a review really of what what I've covered in the pre-recorded sessions. So hopefully you've been able to see those, and now you have an opportunity to do a bit of a Q&A if you wish to. You're welcome to ask any questions. You'll see there's a Q&A option. So if you have any questions that have arisen as a result of some of the recorded sessions that you've seen, or indeed anything that comes up tonight. So my plan this evening is just to go through the essentials of the topics that have been covered already, a bit of a review. Some of the tasks that I do today will be ones you've seen before, but it's a good opportunity to recap on those things that I've done already. So first of all, a little recap of what it was that we were trying to cover during those recorded sessions. So this is the topic here was cells and cell transport. So the first recorded session was all about cells, where we looked at different parts, the functions of the names of and functions of the different parts of the plant and animal cell. We looked at specialized cells or differentiated cells, and then the levels of organization within organisms, so from cell to tissue to organ systems into organisms. And then the second half of this unit then, and the second pre-recorded session that you hopefully have seen, is diffusion, active transport, and osmosis. So I went through what diffusion was, how it works, factors which affect diffusion, and then looked at active transport, which kind of works in the opposite way to diffusion, and then the special case of osmosis. And really, as you see these on here right now, that's a very much increase in difficulty from top to bottom. Osmosis would be deemed one of the more difficult topics at GCSE, and I think parts of plant and animal cells, certainly naming the parts, is very much the low end, certainly a foundation sort of question, foundation tier question. So that a lot of that will be stuff you've covered before. So let's go through some of those key things that I went through before. So remember that in our animal cells, we've got these, well, four essential features. I would probably say the three essential features. We've got the cell membrane, the nucleus, and the cytoplasm. And if you recall, I asked you to try and remember what these different parts of the cell were. So you can see this is where I asked you to pause the video, and you're more than welcome to do that or to try and do it quickly yourself now, and try and identify these different parts. So can you think now what number one was? Number one was the nucleus, and number two, can you identify that thing on the outside? That's the cell membrane. And number three in the middle there, that's the cytoplasm. And four then is certainly, that's more a GCSE concept, this idea of a mitochondria. So there's a naming of functions. They're quite distinct features. They're quite obvious identifying those. Cytoplasm is that. Well, hold on. Let's now look at the function. So the nucleus, what does it do? So we just say it controls the cell's activities. The cell membrane controls what comes in and out of the cell. Okay, the cytoplasm is that jelly-like substance within the cell where all the chemical reactions take place. And the mitochondria are where respiration takes place. Respiration is the second topic, second unit then, should I say, of GCSE. So you will probably have covered it by now, but at this point, this is the first unit of GCSE, respiration is you're relying on what you've covered in previous years, but you certainly will have covered it. So what is it? It's the release of energy from food. Now, if I put those numbers back up, what if you can identify what those are again? So number one, controls the cell's activities is what the nucleus does. Number two, controls what comes in and out of the cell. And number three is where chemical reactions take place. Number four is respiration, where respiration takes place. And I've said respiration. Number four is the release of energy from food. Lovely. Plant cells. Well, look, we've got the same sort of thing going on. Can you identify numbers 1, 2, and 3, and 4 here? Well, 1 is the cell membrane, just as we've seen before. 2 is the cytoplasm, just as we've seen before. 3 is the mitochondria, just as we've seen before. And 4 is the nucleus. What's different in a plant cell? So you can see here now that obviously looks different to an animal cell, but there are some extra features on here. we'll use this as a good opportunity to recap again. So what are those different parts? We identify number 1, that's the chloroplast. Number 2, the cell wall, and number 3, the vacuole. And what do they do? Key features here: cell wall is all about strengthening the cell. It's made up of cellulose. Chloroplast, remember those sites of photosynthesis, and they got this pigment molecule called chlorophyll, which absorbs light, and then the vacuole stored in cell sap. Okay, I'm going quite quickly because I know you've seen this before, so we're just using this as an opportunity to recap. There we are. Can you remember those things on there? Number 1, what's the cell wall made of? It's cellulose. Number 2, chloroplast, site of photosynthesis. Number 3, that molecule is chlorophyll. And number 4, stores cell sap. We then went and looked at specialized cells. Remember what the idea of specialized cells is: that the cells that we show as our basic cell, like the ones I've just shown you, particularly the animal cell, not many cells really look like that because that's our basic cell, but cells undergo this process called differentiation, and that is a process which makes them specialized to do a particular job. Examples are things like sperm cells. Well, you know, you can take your pick. You can, for any part of the body, a heart cell, a muscle cell, a liver cell, a brain cell, or a nerve cell, or a sperm cell. Some of them look absolutely distinct. The sperm cell being one, nerve cell being another, and some look, you know, pretty similar to each other. But what we do know is that when we get a group of similar specialized cells, which have this similar function, we call it a tissue. And if we got 2 or more tissues that perform a specific function, we call it an organ. And then when a group of organs work together, we call it an organ system. And lots of organ systems work together to form an organism. And that's that sort of hierarchy, starting with the cells, and it's all about collectively having a function. So a group of cells which are specialized in a similar way, we call tissues, which work together to form organs, and so on and so on and so on. Okay, so that was our specialized cells and our that hierarchy of levels of organization within an organism. Can we remember those things rather quickly? Number 1, cells. Number 2, tissues. Number 3, organs. Number 4, organ systems. Number 5, organisms. This act now of trying to recall as I go through this is useful, I hope, because you're having to work a little bit and be a little bit less passive and less just listening. You're involved a little bit more. So this was the whole thing put together. Cells undergo a process called, that was differentiation, if you recall. In order to become specialized to do a particular job, it was a group of specialized cells with the similar function is called a tissue. 2 or more tissues that perform a specific function is called an organ. Remember that using that term function is important. A group of organs that work together in carrying out a function is called an organ system, and all the organ systems work together to form an organism. Right, okay, so then we moved on to diffusion. So that's the cell stuff done. We moved on to transporting cells then. So we had, we looked at diffusion. If you recall, diffusion is pretty straightforward. It's just, say, the spreading of particles. I often say it's the movement of particles only. That's fine, from higher concentration to lower concentration. It's the one that makes sense. We're starting with, like it's showing dye in this glass here, that's at high concentration to begin with, and we give it enough time. Those particles will spread out. They move from where they are at high concentration to where they are at low concentration until it all evens itself out. Shown at a molecular level here, where you put a high concentration of dye molecules into water, and they over time they spread out until they're evenly spread. When we do that, we say particles move down a concentration gradient because they're going from a high concentration to low concentration. And this is all passive. I just mentioned the term passive, actually, didn't I? To say that we don't want you being passive in your when you're listening here. It's good if you can be more active, because you're then involved a little bit more and you're having to think a little bit harder. But passive, by definition, means it doesn't require energy. So 2 key molecules that are involved in diffusion that we talk about a lot are oxygen, which we know we need for respiration, and carbon dioxide, which is a waste product of respiration. Oxygen diffuses into the cell, carbon dioxide diffuses out. Or if you look in the lungs, we see oxygen diffuses from the alveoli in the lungs into the blood. and carbon dioxide diffuses from the blood into the alveoli. So diffusion happens all the time, whether going into cells or going happening in the lungs is obviously important too. Diffusion is affected by a couple of things. If you increase the temperature, molecules move faster, and therefore that makes diffusion happen faster. And if you increase that difference in concentration, that will also make it happen faster. So let's look at now, move away from a passive process and go to an active process. So active transport is not passive. It moves the molecules or particles in the opposite direction to diffusion, from low concentration to high concentration. So you can see on the inside of the cell, there's a lower concentration of green molecules. Outside the cell is a higher concentration. So if you want to personify this, those green molecules want to come into the cell. That's what diffusion would encourage them to do, but they can't because they got the cell membrane here, so they're not able to come in. But active transport involves some sort of carrier, some sort of protein, this brown blob that we're seeing right here within the middle of the cell membrane. And the use of energy, because it's causing those molecules to move against where they want to go, from high to low. Instead, this time they're going from low concentration to high concentration. So that involves energy. So it's not a passive process, it's an active process. Right, so let's have a look at these and see how many of these you can recall. So diffusion, the movement of molecules from an area of something concentration to an area of something concentration. So this is knowing it's from high to low concentration. We use this term, they move down a concentration gradient, is 3 on there. We say it's a passive process, number 4, because it doesn't require energy. By contrast, active transport moves from an area of low to high concentration. And it has to use energy in order to pump the substance up or against, meaning the same thing, a concentration gradient. Right, okay, so let's have a look at the last one I want to refer to here, and the hardest one, and probably the one we end up spending most time on, which is osmosis. So osmosis, then the definition here is key. When I teach my pupils this, I say the absolute number 1 thing you need to do is learn the definition for osmosis. There's often a 3-mark or 4-mark question on osmosis, and if you just wrote the definition and had no idea what was going on, you'd pick up 2 marks for sure. So just having that alone is worthwhile. The definition you choose does vary depending upon well, where you look, I suppose. My favorite definition, so there's 2 on here. We can say it goes from a high water concentration to a low water concentration. A lot of teachers like to teach like that. And you'll see that in a lot of textbooks, and that's okay, and it works quite well in the sense that it's just like diffusion, isn't it? Except it has to be water. My problem with it is that you have to always say water concentration. You cannot say osmosis is the net movement of water from a high concentration to a low concentration. That's misleading. You have to say high water concentration to low water concentration, and that causes a lot of problems. So I prefer to use a different definition. I put this on here first. The second part, or the last part of the definition, is always to say across a partially permeable membrane or a semi-permeable membrane. Okay, but the other alternative in that middle bit is you can say that osmosis is the net movement of water from a dilute to a concentrated solution. Now I prefer this because I think students understand what is meant by dilute and concentrated, because they know what dilute squash means and concentrated squash means. So I think this is better. It doesn't really matter which one you choose, as long as you get it right. You know, you get you stick with it. What you can't do is mix up your definitions of diffusion and osmosis. So I prefer to do the green one in there. And so you'll be familiar with this. So which direction is water going to move? Well, it depends on your reference point here. Look, I've said on the left-hand side is a high water concentration. Here, the blue is meant to denote water, and the red is meant to denote some dissolved molecule that's in there. So yes, the left-hand side is a high water concentration, and the right-hand side is a low water concentration. But that is working against this idea of immediately, if I said to you which has got a high concentration, we'd immediately go to the right because we're seeing all these red molecules there. Well, they do. It's right that the red molecules are at a higher concentration, But you see where the problem lies here now. So I said this last time, and it's important to emphasize this. My preference is to refer to the left-hand side as being a dilute concentration. Looks like a weak blackcurrant squash. And a concentrated solution look like a concentrated or a strong blackcurrant squash. So I prefer to use those definitions, but as long as you stick to your one and you make sure you get it in its entirety, it doesn't matter which you choose, you can decide on that. Either way, same thing is going to happen. It doesn't really matter. It's always a movement from high water concentration to low water concentration. So in this case, left to right, or dilute to concentrated. It's still left to right. The direction the water moves does not change. It's just about how you phrase it that you need to get right. And that's what we end up with then, that the water moves from the left to the right, and you can see the level of water has increased. A couple of salient points here regarding osmosis. The effect of cells on, or the effect osmosis has on cells. So when plant and animal cells are put into a concentrated solution, what happens is water moves out of those cells and they will shrivel up. In plant cells, when they shrivel up, it's got a name. We call it plasmolysis, because that cell membrane, the yellow outer part of the yellow part of this this blob, shall I say, that's the cell membrane. That's come away from the cell wall. Normally it would be pushed up against the cell wall, but it's not. That's plasmolysis. That's plasmolysis. If we go the opposite, so what about if we put plant and animal cells in pure water? Okay, so there'd be a very dilute solution outside. Well, that causes water to move into the cell. And the problem with that in animal cells is that water continues to move in and continues to move in, and then those cells burst. The same thing could be said for plant cells, except that plant cells have a cell wall, and consequently it starts to swell, yeah, and it starts to expand a little bit, but the cell wall resists that and it stops them, those cells from bursting, and that situation we refer to as a turgid cell. Now, there's a lot going on here, but we're pretty much done. So let's have a little look at what we should have on here. Osmosis, the net movement of water from a high water concentration to a low water concentration across the semipermeable membrane. We can also say it's the movement from a dilute to concentrated. I say remember, that's my preference. It doesn't really matter what my preference is. It's what makes sense, make most sense to you is the version you need to learn. I sometimes question whether I should give both versions. I don't know. I think pupils probably would just rather I tell them one way. But pupils, textbooks will often say the first version, high water concentration to low. But I don't think pupils really understand that very well. So I prefer dilute to concentrated. So when put in a concentrated solution, is number 5 there. Cytoplasm will shrivel due to loss of water and the cell may die. In plant cells, this has got a special name. We call this plasmolysis when it pulls away from the cell wall. In an animal cell, it's put in pure water. That means that the water outside is going to be much more dilute than inside, so the inside is more concentrated, and so water continues to move in, and eventually that animal cell will burst. Plant cells don't do that because they've got a cell wall which prevents it from happening, prevents that from bursting. And we say that that a plant cell in a situation like that is number 9. We say it is turgid. Okay, so that is the end of my recap. I hope you found that useful. I'm now going to hand over to Miss Kavanagh, who's going to do a little bit of a recap on the topics that she covered. So we'll just cue this up, I hope, and we'll do that changeover now. Here we go. Go. Okay, Ms. Kavanagh, if you want to present your screen.
Hello everyone. I think hopefully you'll be able to see me. Okay, so we will just run through what we looked at in our sessions, which is the heart and photosynthesis. So first of all, let's have a look at our objectives here and our outcomes for our first part, which is looking at photosynthesis. So we're looking at how we measure the rate of photosynthesis in this section. We're also going to look at the facts that affect the rate of photosynthesis, and to then finally apply our knowledge of photosynthesis to interpret the demands of some exam questions. So we'll have a look at some questions a little bit later in this session. So,
Ms. Kavanagh, sorry
yes.
to interrupt. I'm seeing your. I'm not seeing your. I'm seeing your slideshow, but not in its presentation mode. Sort of, you can present it. So either, well, are you able to start that slideshow?
So I can see on my side that I've got the presentation plus my image there, my video there.
There we are. That's better. I can see that now. That's working.
Okay.
Oh, hang
perfect.
on. It's gone again now. Hold on a moment. Let me see if I can put that over. Well, that looks all right. What happens if you start that now?
Is that okay? I'm not sure if Miss Jones is forwarding it for me.
Oh, right. Okay.
Here. But
Right. Well, it looks good to me now. Anyway.
You can see me in the presentation.
Yeah.
Okay. I think that's on our Carl M. Cymru colleagues.
Right.
Screen.
Lovely. Okay. Over to you.
Okay. Brilliant. Thank you. So we've looked at our first slide there. So we're going over the photosynthesis and the heart. And then we've just talked through our second slide, which is our objectives. First of all, looking at photosynthesis and what we're going to cover in this first part. So we've mentioned in our live session previously looking at the keywords and how important they are and how important it is to make sure that we have a handle on those keywords and we use them as we should do. I'm just going to pause for a second because I don't think my presentation is moving on at all.
No, I agree. I've not seen it move on.
Let me just have a try for a second.
That's better.
Okay, so if I just bear with me a second. Okay, so Mr. Cobb, can you see my keywords now?
Yes, I can.
And then if I just press photosynthesis, has that moved on?
Yeah, that's it. Brilliant.
Perfect. Okay, so let's run through photosynthesis and remind ourselves what photosynthesis is. So if we remember that term photosynthesis, we can split it up and remind ourselves that photo means light and synthesis means to make. So we are using light, or plants are using light to make what they need. And they put that together and that gives them their nutrition, enables them to grow. What is important is that you remember the equation for photosynthesis, and that's the word equation that you need to remember. So we start off with the things that react together, our reactants. So we've got carbon dioxide plus water, and then we add that light energy and chlorophyll to enable that process to take place. And remember, Mr. Cobb has talked to us about plant cells, so we know that this takes place in the chloroplasts in the plant cell, and that gives us glucose, which is a sugar, and it also gives us oxygen, which is a gas. Then that gas of oxygen is released through the stomata on the underside of the leaf, which we will talk about later. Okay. So leaves, we know, have a particular shape that enables them to be adapted to enable maximum light absorption, but to also enable photosynthesis to take place efficiently. So we can see these two examples here, and that reminds us that they are thin, they are wide and flat leaves. We've also got an extensive network of veins that enables the transport of substances, whether that's water or whether it's solutes around, and also the stomata, and we'll have a look at those in a little bit more detail on another slide. So here we've got the structure of the leaf, and this image is showing us inside the leaf and how complicated it might be, but also how important it is that we remember all of these parts and are able to label this, especially if we are following the separate science route. So we've got the upper epidermis here, so the top of the leaf. What's important here is the palisade layer. So this layer here of our plant cells that are elongated and tightly packed together. So they're elongated, so their size is long, and that enables them to have a great number there. You can see they have many chloroplasts in their structure, but they are also tightly packed together, so there is not much space between any of these cells here, and that enables the maximum amount of photosynthesis to take place when that sunlight is shining on the top of the leaf there. As we move down and we look at the spongy layer here in the middle of the leaf, we can see that there are still cells here. They still have chloroplasts, so photosynthesis will still take place in this middle section, because any light that doesn't quite get through those top two layers will hit those chloroplasts in that spongy layer there. But you can see that there are gaps here, and there are many more gaps than what we've had previously in the other two layers, and that enables gases to move freely in the leaf. And we remember that carbon dioxide enters the leaf, and that carbon dioxide enters the leaf here through the stomata, and then oxygen and also water vapour can leave through the leaf, through those pores there on the underside of the leaf. So that's the spongy layer, so more room here for gases to move and to exchange. Also, we may find something called the vascular bundle, and the vascular bundle contains xylem vessels, and also phloem. And it's the xylem that transports our water, acting quite like a drain pipe that transports water throughout the plant, and then our phloem over here that transports our sucrose and our dissolved sugars to enable those to move, either if the plant needs to store them or if it needs them in any processes, for example in respiration. At the bottom here we've got the lower epidermis. As you can see, as we move down the leaf, so from the palisade layer through the spongy layer to the lower epidermis, there are still some chloroplasts in the lower epidermis, but they are fewer in number. So photosynthesis can still take place here, but it's at the top of the leaf where that maximum photosynthesis takes place. So this is something that Mr. Cobb has mentioned, but I feel it's worthy of just a quick revisit. Here's our plant cell with the vacuole, the cell wall, the cell membrane, mitochondria, nucleus, cytoplasm, chloroplast, and the vacuole. So just a reminder that this is the part, the chloroplast, that we are interested in photosynthesis. That's the site of photosynthesis and where that takes place. And we mentioned glucose and how important glucose was. If we remember from the previous session, glucose is a sugar and it's produced by the plant, and it's used in a number of different ways. It's used in respiration as well for the plant, and it can be stored as starch, but also what is crucial is that it's turned back into glucose at any time when the plant needs it. Okay, so here we've got a reminder of some of the key terms that we've looked at so far. and some of the key relationships really in terms of these words and their meanings. So the stomata, if we remember, the stomata allows gases in and out of the leaf, and they are surrounded by cells called guard cells, and it's that movement there that enables gases to come in and out, so gas exchange, but also enables water to leave the plant as well. Chlorophyll there absorbs light, so chlorophyll is found within the chloroplast, and then our leaves need to have a large area to enable light to be absorbed. Our veins transport and support the leaf and the plant, and then finally having a thin leaf allows gases to reach the cells very quickly and efficiently. So we looked in our other session about this idea of limiting factors, and there are three main limiting factors that can affect how well plants photosynthesize, and they are light intensity, temperature, and carbon dioxide. And there are some graphs that you need to be aware of and you may well meet in your exam, so you need to be able to interpret these and understand what is happening here. So in this purple line here we can see that the rate of photosynthesis is increasing, and it increases up to a certain point, and then the graph starts to level off. And that means that another factor is causing photosynthesis to slow down and to not take place. Similarly with carbon dioxide, the rate of photosynthesis increases up to a certain point, and then after that the rate of photosynthesis levels off, and that means that a factor is preventing that from taking place. Our temperature graph is slightly different. So for our temperature graph, what we need to remember is that photosynthesis is regulated by enzymes, and as we can see here, the shape of our graph is different. So as temperature increases, so does the rate of photosynthesis in this first part of the graph, but only up to a certain point. And then after that certain point is reached, which is called the optimum temperature, after that point, our rate of photosynthesis slows down and declines. And that's what you can see there, where the green box is surrounding that. And that is because we've mentioned that an enzyme catalyzed reaction takes place in photosynthesis, and that means that it's slowed down, our active sites have been damaged and distorted because the enzymes have been denatured, and that means our reaction can no longer take place. So briefly, we know there are three limiting factors. We know an increase in light will increase the levels of photosynthesis, but only until a certain point when another factor becomes limited. In the same way, increasing carbon dioxide levels will increase the levels of photosynthesis up to a certain point, and then another factor comes into play. Photosynthesis is a chemical reaction, it's true. Then we looked at a standard practical, so this is a pondweed investigation. So our pondweed is placed at different intervals away from a light source, and then we measure the numbers of bubbles here of oxygen given off. And you may be asked to plot a graph, and we can see here just a reminder of the equation. So here's some data. You may be asked to plot a graph and then analyse that, and our data tells us that as we move away from our light source, then our number of bubbles decreases. And just a reminder here of, if you are drawing a graph, what you might need to include. I'm just going to run through this because we want to have a look at the heart as well. So this presentation will be available for you to have a look at a later date. We'll just have a look at an exam question then to do with photosynthesis. Quite a few exam questions will ask you to include the word equation for photosynthesis in some form or another. So here we can see for 2 marks here, our first question is asking us to complete the word equation for photosynthesis. So we need to add, first of all, the two parts to this equation here, something plus something, that's worth 1 mark. And then glucose plus something, so that other something there is our second mark. So here we're looking at carbon dioxide plus water, gives us glucose plus oxygen. Okay, and if you did include the symbols, then the examiner would credit you for that. In the second part here, this is a 4-mark question. We've got a leaf from each of the plants here that were tested for starch using iodine solution. So a reminder there that those chemical tests that you might have done perhaps in digestion and as well in photosynthesis, looking at iodine, testing for starch, but also Benedict's testing for glucose or sugars, then that will be important here in this section as well. So plant X had been growing in the light before it was tested, and then plant Y had been taken from the light and placed in the dark for 48 hours. And we need to explain those results here that we see where we've got the blue-black colour, the pale brown colour. So for 4 marks, so it's about a small description and then an explanation. So plant X has photosynthesised. And we know that because plant X has got starch, and we know it's got starch because the iodine has turned the blue-black colour. Remember, iodine starts as a reddy-brown colour. In leaf Y, we've had no photosynthesis that takes place. And we can see that because we know that that leaf there has returned a pale brown colour with iodine, and therefore any starch has been turned into glucose, it's been used up, or it's been destarched the plant. So we know that there is no starch there. So that test for starch was negative. Okay, so here we've got a leaf structure question. And again, that's something that we need to be familiar with. We need to label A and B on this part here, which we saw earlier. So A is the palisade layer and B is the spongy mesophyll. And then the process, our second question here, which occurs in layers A and B, is photosynthesis. The function of phloem enables us to carry sugars around the plant. And then our sentence here that we need to complete, sugar is used by plant cells in the process of respiration, or it is converted to starch for storage. And then one function of the stomata, our final question here, is that it controls the amount of water that enters or leaves the plant, or allows gases to move into or out of the plant. And again, gas exchange would be useful there as well. So I'm going to move through now to look at the heart, which was our other area that we looked at in this section. So we were looking at how the heart is supplied with blood and how blood flows around the heart and around the body. So looking at the circulatory system. We were also looking at labelling parts of the heart and to look at that journey that blood takes through the heart, thinking also about the function of valves and to explain that term double circulatory. So if you remember, we looked at the blood vessels and what their name is that supplies blood to the heart, and that's the coronary artery. Then we looked at what the heart muscle needs from blood, and that is oxygen and glucose. And then finally, why does the heart need that oxygen and glucose? And it's for the process of respiration. And part of this section to do with the heart, we need to look at blood vessels. So we talked about the structure of the artery and the vein. And you can see there how important the structure of the artery is, because blood is flowing through the artery under high pressure. So we need to make sure that it is very strong there, but also elastic. As we look at the vein, we can see that the vein has got valves, and this stops the backflow of blood. And we'll talk about that later when we look at the heart as well. So arteries, strong and elastic layers to enable the blood to flow under high pressure. And we can see there the aorta, which is an artery, a very, very strong and important artery that takes the blood from our heart around our body and you can see how it leaves the heart there. Circulatory system, we can see here we've got arteries and veins that show the pattern that blood travels in. So oxygenated blood in the red, and then deoxygenated blood in the blue. So that returning that carbon dioxide so it can be removed from our body. So here we've got a diagram showing the veins, and we can see they've got valves here. So in the middle here we've got valves, and as the blood is travelling back up towards the heart, those valves close like a little doorway and stop the blood flowing backwards and pooling in one part of our body. So that enables it to reach back up to the heart when we need it to. And we can see that here in these two diagrams here. Capillaries are a third type of vessel, so very, very tiny blood vessels that surround an organ generally, but we can see here that they are 1 cell thick, very, very tiny vessels that enable transport of substances around the body. So moving on to look at the structure of the heart. So we can see here this external view of the heart. We can see how we've got the vena cava, which is this vein here. We've got the aorta here, and then we've got the external structure with all of our blood vessels here as well. Inside, so slicing that heart open, we can start to begin to see some of the structures of the inside of the heart. So we can see the main artery and vein on the outside of the heart, but we can also see the ventricles, the atria and the ventricles. We can see the septum dividing the two sides of the heart, and the walls here of the heart. Remember, the heart is made of something called cardiac muscle, which is very strong. It also never gets tired. And we can see here that one side of the heart is thicker than the other side. So that tells us here, this thicker side of the heart is able to pump blood around the body, whereas the thinner side of the heart is only pumping blood to the lungs. And that reminds us of what we call the double circulatory system. So we've got one system that is pumping blood to the body, and then a second system that is pumping blood to the lungs. Okay, so this diagram shows us some of the labelled parts of the heart, and that's something that you need to be familiar with and to remind yourselves of, and learn all of these labels and also what happens in each part. So you can see we've got oxygenated blood here in the left side of the heart where it's shaded in red, and then deoxygenated blood here in the right hand side of the heart. We've got the aorta that's taking blood around the body. Remember, we've seen that artery structure that's very elastic and very strong, that enables that blood to be transported very easily around our body. We've got the pulmonary vein that is bringing oxygenated blood into the atrium here from our lungs. It then travels through the left atrium, through our valves here. So those valves stop that blood flowing backwards. So through the left atrium, into the left ventricle, and then out into the aorta around our body. On return, then we've got deoxygenated blood that comes into the vena cava here and here, and then enters the right atrium and then the right ventricle, and then it leaves through the pulmonary artery, and that deoxygenated blood is taken to the lungs where it picks up oxygen, and that whole cycle, that cardiac cycle, begins again. So we can see that here, that we've got oxygenated blood coming in to our left ventricle and deoxygenated blood coming into our right ventricle here. And then as the valves open on both sides, those different types of blood come into our left and right ventricles, but there is no mixing of that blood here because of our septum, that wall between the two sides. As that blood then enters, whether it's oxygenated or deoxygenated, it is then able to move up and out. So either around our body or to our lungs. And then we can see here a very stylized structure of our circulatory system. So we can see the pulmonary artery, we can see the vena cava here, we can see the pulmonary vein, and we can see the aorta. So these are things that you need to make sure that you are familiar with to ensure that you can label these on an exam question. Also, what we need to have an awareness of is cardiovascular disease. So we can see here the buildup of an atheroma inside the artery, and that prevents oxygenated blood getting through as fast as we want it to, and that could lead to a heart attack or a stroke. To treat that, we could insert a stent, and you can see here how that increases the area of the artery. So we're just going to have a look at one question just to summarise what we know in terms of the heart. So here, this question is asking us one substance that is transported by blood in the circulatory system. So 1 mark here to respond with either glucose, oxygen, carbon dioxide, or products of digestion. There's quite a range of suggestions there that we could give. And then our second question is asking us to, again for 1 mark, to describe what the main tissue is in the heart wall, and that is cardiac tissue. So this presentation will be available later on in Carlam Cymru for you to have a look at, and there are extra questions here at the back if you want to have a look at that. But just like to take this opportunity to say thank you for your attendance tonight and for watching this session, and also wishing you the best of luck for the future. Thank you.